This presentation is designed to show you the key differences between mitosis and meiosis. First of all, we have to realize that these X's over here and on the meiosis side are actually replicated chromosomes. They're identical strands of chromosomes that have been already replicated in DNA replication earlier on. So this strand and that strand are identical. These two are homologous chromosomes, and in mitosis, all the chromosomes line up in the middle equator, one after the other, all lined up uh, as such. In meiosis, in metaphase 1, the homologous chromosomes, which were lined up over here, one after each other, are now lined up, wrapped around each other. I call it hugging. They're all wrapped up. And this is a key point for genetic variation, because when they're all wrapped up, very, very similar portions of each chromosome are touching each other, and it's very easy. These chromosomes are kind of fragile, and it's easy for them to become detached, segments to become detached and cross over to the other chromosome. So you're getting a lot more mixing of genetic material when they can cross over like that. You might get a blue eye, blonde hair genes which are really close to each other and have always been part of a population together. Suddenly now, because of crossing over, the blue eyes might end up with brown hair in another generation. So the crossing over increases the variability and the mixing of genes in a population. But here we have everything lined up, and in mitosis, what we're going to see now is the chromosomes are going to separate, the chromatids are going to separate and go to opposite sides of the cell, and then a new cell will form, and we will end up with two cells that are identical. No, it's to the original cell, with 1, 2, 3, 4, 5, 6, 7, 8 chromosomes in each. That's gone, and now we end up with two identical cells. What happens in metaphase 1 is the homologous chromosomes separate, one to one side, one to the other, and two new cells form. Which have half the number of chromosomes in them as originally in each. And so we have created the haploid number, half the number of chromosomes in each cell. This cell has 2, 4, 6, 8. This one has 1, 2, 3, 4. Now don't forget, these are identical copies in each case and will be separated in the next step. So in mitosis, now we're done. We've copied. We've gone from 8 chromosomes in one cell to 8 chromosomes in two cells, identical chromosomes. But in meiosis, now we're only half done. Now we have, in meiosis, in metaphase 2, we have two cells where the 4 chromosomes are lined up, again, along the equator, the middle part. And now it's time to go through that division that was very much like mitosis before. The identical chromosomes, chromatids, are separated in each cell, move to opposite sides. The old cell is gone, a new cell forms. And we have two identical, or two cells with half the number of chromosomes. Same thing goes on in the other side. And we have chromatids separating going one way and chromatids separating and going the other way. The old cell disappears. And again, we form up two new cells. This is happening all at the same time. And we end up with 4 cells with half the number of chromosomes. These gametes, these sex cells, have 4 chromosomes in them. Whereas, if you recall, in the beginning, the cells had 8 chromosomes. So the key to getting the reduction part of this meiosis is in meiosis 1, the chromosomes do not line up one after each other like here. They line up, the homologous chromosomes are formed together, wrapped up around each other, and then they will be separated out as such in the first division.